So it's good to see that you have all decided to come back after coffee, which is always the first challenge. Okay, so I think it's pretty clear that over the last 60 years, pretty much the paradigm of evidence-based medicine has been this concept of what I would call one regimen to rule them all, a standard of care, a control which simultaneously maximises good outcomes, minimises bad outcomes as best we can, is recommended in guidelines and should generally be used in all or most patients. And it's iterative improvement to this optimal regimen through a series of what have generally been two-armed trials versus the current SOC that has substantially improved outcomes in a number of areas. What we've spent most of today talking about so far is the innovation over the last 2 decades, which is really focused on comparing multiple new interventions or different durations versus SOC to iteratively improve SOC faster. And of course, when we choose SOC, we can think about guidelines, we can think about clinical practice, but the practical trial design is really predicated on this question, is there actually always a SOC? And in particular, what do we do if there isn't? And the motivating example is rising antimicrobial resistance, which is leading to increasing numbers of multi-drug resistant bacterial infections, particularly in areas of the world such as Southeast Asia, and also to stronger and stronger antibiotics being used empirically, meaning this problem isn't going to go away. But because this is a new problem, there are lots of questions about how we should treat these severely drug-resistant infections. They do generally have high mortality and morbidity. And there's a class of drug called carbapenems, which were previously considered to be the last resort. And it's one particular type of bacteria, Enterobacteriaceae, that are resistant to these carbapenems, so-called CPE, that are a particular concern. So if we specifically think about CPE infections, there have been some new or newer antibiotics over the last few years. You know, could they have a role? They're often quite specific. One question is actually whether we've got the dose right. So even though, at least in theory, these bugs are resistant to carbapenems, could we actually just give higher doses of them to overcome low-level resistance? There are some old drugs like this drug polymyxin B. Could we think about using drugs like that, plus or minus other drugs which might synergistically increase their potency, and zidovudine is an option? What about combination therapy? So could we combine 2 different carbapenems? But then, of course, once you start talking about combination therapy, you can just add to this list forever. So everything plus everything else. And there's 13 things there, and there will be more that people have thought about. So why not use platform trials? Why do we need even more innovative designs? So I've just shown you there are lots of antibiotics and lots of durations even with a very weak evidence base, but very strong opinions from clinicians about what they would want to use, meaning it's extremely difficult to identify what is SOC. But actually in antibiotics, it's even worse than that, because even if you do get a group of people to decide what they think SOC might be, for many patients, they just won't be able to take it. And that's for many reasons. The first is that the resistance that we worry most about is generally mosaic, cross-class. Now, what I mean by that is the resistance genes aren't actually on the bacteria itself. They're on separate bits of genetic material which move in and out of these bugs at really frightening speeds. So just because you're resistant to one antibiotic doesn't mean you're resistant to the other. You get this massive mosaicism. Many antimicrobials also have got really important contraindications, and many people with bacterial infections are old and comorbid. Even worse, we actually never find the pathogen in many patients, so we don't even know whether they really had a bacterial infection. So should we actually be comparing stuff versus nothing? But actually, again, a lot of these patients will die whether or not you find a bug. That's outside complicated UTI, which makes it very hard for a clinician to randomise against something which they feel might be a substandard SOC. And actually, it's even worse because for antibiotics, resistance means that actually we need diversity. One SOC will just drive resistance to that drug. But that means we need multiple trials against multiple non-existent different SOC, which many clinicians won't randomise to. Many patients won't be eligible to receive for multiple reasons, leading to results which are simply not relevant to most of the patients, and results which accumulate very slowly and potentially have low power to exclude important differences. So, in a practical design, there is just no SOC. It's very simple. Each patient is randomised between a subset of regimens, and these subsets differ from patient to patient. In contrast, in the traditional platform designs that we've talked about this morning, there is still one SOC, which every patient has to have the potential to be randomised to. We do indeed test multiple intervention arms. We can start or stop. The SOC can change over time. You don't have to randomise to all test arms, but this primary analysis, as people have talked about throughout the day, is still a pairwise comparison with SOC in patients randomised to SOC versus each test arm. So going back to my example, here are my original 13 regimens, and there are just 8 example patients across the top, and these would be typical patients presenting with these serious CPE infections. And yes, no just means, would you actually even consider one of these regimens in that type of patients? There is no regimen with yeses in every column. So instead, what would happen in a personal randomisation list is we consider for each patient what regimens might be relevant for them, and we randomise to those ones. It answers the clinical question, out of what I could use for this patient, what is going to give me the greatest chance of success? And that's because how the things that you could give this patient might compare to something else that this patient can't take is completely irrelevant, whether or not it's someone else's SOC. Now, of course, there's a trade-off, yeah? And that is really that you have to be able to argue that given the number of different options and the lack of SOC, choosing a treatment which is likely to be one of the best out of the multiple available options for that patient is more important than identifying the very best treatment, which is the type 1 error paradigm. What we really want to do is just avoid the worst. And so if you think about putting, an efficacy outcome like mortality on the x-axis and another outcome like resistance or cost on the y-axis, the SOC paradigm is identifying the very best treatment, assuming that the vast majority of patients can take this. Whereas the trade-off with the practical design is what we really want to do is work out what's the worst thing for each patient and avoid that. Meaning that if you have a new patient who can't actually take D, you still have information about how you should treat them. Implications, no frequentist testing, all the Bayesians say hurrah. Sample size is determined by information gained versus the current status quo. Becky will talk about that next. But I would argue that the current trials that we've got are really not designed to answer the question we need answered, which is what are the best regimens out of the available options for multi-drug resistant infections that will reduce morbidity, cost and mortality overall. And that's because there's no SOC, and practical is a design to answer that. Thank you.